When a patient takes a conventional drug, it does not go straight to the disease. The medication saturates the entire bloodstream, flooding healthy organs and causing collateral damage just to deliver a small fraction of the active dose to the actual target site. Nanomedicine addresses this problem through targeted delivery, using engineered carriers to transport drugs directly to the site of the disease. This discipline operates exactly at the nanoscale, dealing exclusively with materials engineered to measure between 1 and 100 nanometers in diameter. To put that in perspective, 1 nanometer is exactly 10 to the negative ninth, or 1/1000th, of a meter. Shrinking materials to this specific scale alters their physical and chemical properties. Because these particles are smaller than the natural pores of human cells, they can bypass standard biological defenses and breach cell membranes. Once inside, these artificial structures are small enough to interact with internal cellular machinery, binding to specific proteins or delivering gene-altering sequences directly to the DNA. By utilizing this scale, clinicians can move away from systemic bodily saturation and focus instead on microscopic, site-specific precision. Engineers have designed a wide array of nanocarriers for specific payloads. The current arsenal includes polymeric nanoparticles made from biodegradable materials, alongside metallic structures built from gold or silver. For complex biological environments, researchers built specific geometric shapes, such as highly branched dendrimers or rigid cylindrical carbon nanotubes. However, the most clinically significant structure for targeted drug delivery is the liposome. This diagram illustrates a liposome's cross-section. The yellow outer ring is a phospholipid bilayer, a synthetic membrane mirroring human cells. It surrounds and protects an aqueous core. A single carrier can trap water-soluble drugs inside the core, while embedding fat-soluble drugs within the membrane walls. This capacity to transport difficult-to-deliver chemical payloads directly to a target enabled the development of specific cancer therapies, such as liposomal doxorubicin. Once they are injected into the bloodstream, these engineered liposomes must navigate to a tumor without internal guidance systems. They do this by exploiting the physiological flaws of the tumor itself. As a tumor rapidly expands, it builds irregular blood vessels with wide, leaky gaps between endothelial cells. Because nanoparticles are smaller than these gaps, they escape the bloodstream and enter the diseased tissue, remaining trapped inside the tightly sealed vessels of healthy organs. A second biological flaw traps them there. Tumors have poor lymphatic drainage systems, meaning large molecules struggle to be flushed out. With the exit routes restricted, the nanoparticles pool and build up in high concentrations precisely within the tumor space. This two-part mechanism is the enhanced permeability and retention, or EPR effect. Passive targeting relies on utilizing the structural incompetence of the disease to ensure drug accumulation. While the EPR effect provides baseline accumulation, researchers apply a secondary layer of control through active targeting. This involves modifying the outer surface of the nanoparticles with specific antibodies or ligands. These surface modifications recognize and bind exclusively to specific receptors found on the surface of target cancer cells. This binding mechanism is critical for bypassing the blood-brain barrier, a biological filter that typically prevents treatments from reaching the brain to address Alzheimer's or Parkinson's disease. Ligand-modified nanoparticles can cross this barrier by utilizing a natural transport process called receptor-mediated endocytosis. When the particle docks with the target receptor, it triggers the cell membrane to fold inward and swallow the particle whole. To deploy the payload, researchers use controlled release mechanisms. For example, pH-sensitive nanoparticles are engineered to remain stable in the blood, but dissolve when exposed to the acidic environment of a tumor. Doctors can also trigger delivery from outside the body, using targeted bursts of temperature, light, or magnetic fields to release the drug on command. Integrating active targeting with these external triggers allows clinicians to treat nanocarriers as programmable delivery systems. These mechanisms drive the field of theranostics, the integration of therapy and diagnostics. Using gold nanoparticles, clinicians can image a tumor on a scan and then heat those same particles to destroy the malignant tissue. In genetics, nanoparticles provide the protection required to deliver fragile DNA or RNA payloads for CRISPR gene editing. Metallic variants serve other clinical functions. Silver nanoparticles generate antimicrobial effects, making them effective when used in wound dressings and surgical coatings to prevent infection. In vaccine development, lipid nanoparticles act as the structural armor that protects mRNA molecules from degradation in the bloodstream. This protective mechanism enabled the deployment of mRNA vaccines during the COVID-19 pandemic. Because lipid nanoparticles require specialized manufacturing, constraints in the nanoscale supply chain can halt vaccination programs when stock is unavailable. Nanomedicine is currently integrated into treatment protocols across oncology wards and global vaccine distribution centers. Operating at the 1 to 100 nanometer scale carries risks. The same size that allows cellular penetration can also lead to toxic accumulation in vital organs like the liver, lungs, and brain. This accumulation can trigger a toxicological cascade, starting with the generation of reactive oxygen species within the cells. As this flowchart illustrates, the generation of reactive oxygen species leads to cellular inflammation, which can result in permanent DNA damage. Beyond biology, the field faces logistical hurdles. The high financial cost of development and the engineering difficulties of mass-producing these particles restrict their commercial availability. Health agencies also face a regulatory challenge when evaluating synthetic materials whose long-term effects on the human body are not yet fully understood. While nanomedicine offers the potential for personalized treatments and surgical nanorobots, its current application remains a balance between achieving microscopic precision and managing unforeseen systemic blowback.